The first in Bulgaria scientific symposium on epigenetics and signaling in stem cells was held on 16th and 17th April. The forum is part of the TransStem project. The project aims to unlock, promote and maintain research studies in the field of stem cells at the university and in the region to the highest world levels. The project will implement research developments focused on both fundamental research on stem cells and their clinical application for diagnostic and therapeutic purposes. Dr. Mario Vinciguerra, Air Chair in Translational Stem Cell Biology, presents the event.
Hello, good morning. During the days of April 16 and April 17, the Medical University of Varna hosted the first virtual symposium on epigenetics and signaling in stem cells. This symposium was supported by the TransStem project of the Horizon 2020 program, and it was the first of its kind organized in this city and also in Bulgaria. The program was developed in 2 days, and the first day was about the biology of embryonic and pluripotent stem cells, while the second day, on April 17, the program was about adult cell stemness and plasticity. So the program really grouped and attracted the best minds and experts in the field of stem cells in Europe. And in the next 5 minutes or so, I will explain you briefly, or I will list briefly what were the topic of this interesting symposium. On Friday, April 16, the symposium was opened by a keynote lecture of Professor Luciano Di Croce, who is the ICREA professor at the Center for Genomic Regulation in Barcelona, in Spain, who gave a very interesting overview of the molecular complexes and transcriptional regulation that govern the fate of embryonic stem cells, adult stem cells, and cancer cells. This lecture basically provided an introduction for all subsequent intervention. The following lecture was by Dr. Yasser Atlasi of the Queen University of Belfast. Dr. Atlasi gave a very sophisticated talk about the role of enhancers that are genomic regions that regulate, regulatory regions that regulate the expression of genes, are non-coding regions. And he developed a very interesting research on the role of these enhancers in the shaping the cell signaling and the fate of induced pluripotent stem cells. That opened the way to a series of talks on induced pluripotent stem cells. In fact, then was the turn of Dr. Vladimir Rotrek of the Maastricht University in Brno, Czech Republic, who provided a compendium on hypoxia, the role of hypoxia, and also cell signaling, such as the role of MAP kinase in the metabolic modulation of human pluripotent stem cells. Then Dr. Giancarlo Forte of the International Clinical Research Center in Brno, always in Czech Republic, gave a very interesting talk on the mechanism of mechanosensing. That means that the stem cells are biologically located into a matrix, into tissue, and the force applied to these tissues from the matrix shape also the function and the fate of stem cells. This was very original. After Dr. Forte, it was my turn, and I gave a lecture on the role of histone variants that are epigenetic molecules that substitute canonical histones in the structure of the chromatin, and the role of these histone variants, in particular one called macroH2A1.1, in the repair of the DNA. Because what we know is that when we generate pluripotent stem cells, the cells experience high levels of DNA damage. This DNA damage is important for the subsequent genomic stability of the cells and also differentiation of the cells. So finding ways to regulate this DNA damage response, it will be important. And the first day of the symposium was closed by Dr. Dave Hay. David Hay is from Scotland, from Edinburgh, and he devised in the last 20 years a way to develop artificial livers, so livers in vitro using pluripotent stem cells. This was also very original. Then we went to the second day. It was a Saturday, April 16. There were 5 lectures. The first lecture was from Oksana Bereshenko, a Russian researcher located in Italy, that explained how we can regulate glucocorticoid signaling in hematopoietic stem cells. Glucocorticoids are very useful drugs, old and useful drugs in a variety of disease, also against the COVID disease, for instance. And through generation of genetically modified mice, Oksana and her colleagues demonstrate how glucocorticoid signaling can govern the differentiation of hematopoietic stem cells. After Oksana, our Professor Tonchev of the Medical University of Varna describes how transcriptional profiles are important in regulating the fate of adult neurogenic stem cells in monkeys, in primates. And this is a very useful and original model, and his talk was very successful. After Professor Tonchev, it was the role of Professor Resnick. As you know, he's the coordinator of the TransStem project. He has a lifelong experience in bone marrow transplantation, and his was the most clinical talk. So he gave really an overview of the role of hematopoietic stem cells in curing people by bone marrow transplantation, for instance, in the case of hematological malignancies. After Professor Resnick was the turn of Dr. Gauthier, Benoit Gauthier, a Canadian professor that works in Spain, in Sevilla. And his talk was quite interesting and also particular, because it was not really on stem cells, but it was on transregeneration, the process by which a cell type can transform in another cell type that is completely different. So basically he showed data on the pancreas that demonstrate that with the interplay with the immune system, a cell type like alpha cells can transform into beta cells and vice versa. And this also have a strong implication for the treatment of diabetes. The last talk of the symposium on Saturday was the talk of Professor Tanja Roskams. Is a Belgian professor working at the University of Leuven. Tanja Roskams talk about the liver progenitor cells that are present in the liver parenchyma and are able to regenerate hepatocytes and hepatic tissue once the liver is damaged by, for instance, fibrosis or cirrhosis, liver disease that are induced by an incorrect lifestyle or by viral hepatitis or by alcohol consumption. And then this was the last talk of Saturday, 17th of April. And I have to say that I believe this symposium was highly successful with many questions and also hundreds of people who connected. And that demonstrates that the field of stem cells is very much alive with its applications, but also with its controversies. I believe that this symposium basically exposed the local research community to a variety of topics and research directions that are ongoing in Europe, because when performing research is absolutely necessary to educate about what's going on in other countries, and also provided the opportunity to connect to these researchers. So local researchers, there were several questions, they connected to the speakers, and this can eventually lead also to collaborations. So this will be an opportunity for professional growth.